We're good to go.
Okay, I will share my screen. Can you see my slides?
Yes.
Okay, so first of all, thank you for inviting me. Oops, no. Thank you for inviting me. It's a great pleasure to talk here today.
Louise, just to quickly interrupt you, sorry, we're still seeing not the presentation mode.
Okay, so I think I will share my entire screen actually instead of just the presentation. Yeah, just a second. Better?
Yes, that looks perfect.
Yeah, sometimes, well, it doesn't work pretty well, right? But anyway, so let's restart. Yeah, just thank you. And yeah, today I'm going to talk about my previous PhD, which was kind of, well, the title of the talk is basically what I tried to study in the PhD. So enhanced neural processing of attended stimuli, decoding the content under spatial and temporal attention. And so when we started the PhD, we knew that being able to anticipate events before they happen facilitates stimulus processing. And this is kind of obvious in our daily lives when you are prepared to hit a ball or prepared to do things in the environment, but also in the lab when you consider like a Posner task, when you see that when you have a valid cue that some target will be presented in the screen, in the right screen, for instance, you are more efficient, you respond faster to when there was an invalid cue. So when it didn't, the target didn't appear in the expected position. But little is known about the true effect of attention on the quality of the stimulus representation, because this is special when you consider stimulus that you ignore because you normally the experimenter they don't ask about them. And if you don't ask, there is no way to know how well people are seeing things that they should ignore. In the past, they used it to analyze how well the stimulus was represented, or the quality of the stimulus by the amplitude of the ERPs, the event-related potentials. So you saw that when the stimulus was ignored, you had a lower amplitude of of the evoked potential, and compared to when it was attended. But actually, this is not a good proxy, because even when you try to relate the amplitude of those event-related potentials, ERPs, with the behavior, you don't see a clearly correlation. And especially nowadays with a different proxy of well, how well the stimulus was processed, how well the stimulus was processed in the signal, you can see that there is no really significant correlation between the ERPs and this new tool. And what's this new tool? People nowadays are trying to implement the decoding brain states through EEG and MEG responses. And this is really not really clear right now, so I'm going to show you a picture. So when you show a stimulus to a participant, and actually in an ignored case, you don't ask them about this stimulus. It could be a visual one, for instance, just a oriented bar. And you can show different orientations. And in this case, these different oriented bars are going to activate different patterns across the scalp in the EEG and MEG. So actually, the EEG is really poor at solving this spatial location. So you don't know the source exactly, but with the EEG you can disentangle, you can separate. You don't know exactly where are the sources, but you know that the sources are different because there are different populations processing those different bars. And these tiny differences can be picked up, not by us, not by our eyes, because for me, those two activation maps are really similar, but maybe an algorithm, a machine model could be able to pick this up. So since it's a machine learning, one step that you need to do is to divide the trials you have into training and test sets, because if you do just once, well, let me see if I can disentangle these two bars from those from my electrode setting, then it's not learning. So you need to really separate them. And what you're going to consider is the activation of that part, that different labels, that different bars across your scalp in each electrode. So here you can see that there are two different electrodes, and in red you can see that is the activation or the amplitude for this bar, and in green the amplitude for those channels for this bar. And you can see that this activation in those electrodes are different depending on the label, depending on the orientation that you have. So you can try to separate them by just putting a linear model, just putting like a line to divide them. And what you do next then is, well, let's see if this model can generalize to new trials that my classifier, my model didn't see before. And if it generalizes, it means that, well, you have the information about the orientation in your signal, in this case, in the EEG signal. And how well you can separate them, it will be the proxy of how well this information is coded in the signal, and how well the processing of that representation is, this feature is basically. So we knew that anticipatory states, well, we knew that anticipatory states could modulate how well a stimulus representation could be, but this also depends on the attention and to which domain you are attending, because you have different mechanisms depending on what you're going to attend. So if it's in space, you will have different mechanisms or recruited to induce those anticipatory states related to temporal attention. So we were interested in both, but we knew that they kind of recruit different mechanisms. So in the first study, we will check how spatial attention was modulating the stimulus sensory representation, and the second one, how temporal attention could modulate that, and whether it was modulating it. So in the first study, since it's a spatial attention, and we knew that alpha oscillations are really correlated to spatial attention, and you see a lot of things in the literature showing that alpha oscillations, when you attend to a certain M field, you see that there is a control contralateral activation of alpha oscillations, and actually you have this decrease in activity. And what is alpha oscillations actually? So if you look at any at the EEG activity, you see that this activity across time, it could be translated in the frequency domain, and you can simply describe this time, this temporal function into sines and cosines with different phases, with different frequencies and different amplitudes. And when we consider frequencies, we can discriminate into different bands. And you can see here that alpha is from, I would say, from 8 to 12 hertz or 30 hertz. And you have delta, theta, beta, and gamma. And just to show what is normally seen in the literature, what was the task? One sec. So besides that, you have this contralateral activation of alpha oscillations depending on the spatial domain that you were attending to. You have this also pre-stimulus activity, these anticipatory states related to this alpha power. But in recent years, they were discussing in the literature whether these anticipatory states was related to perceptual bias or a difference in sensitivity, because what alpha oscillations could do, since alpha is normally related to inhibition, and you have this release of the inhibition when you want to attend to that certain spot in the hemifield. You could do two things. If you have just a detection task, it could be that you just are increasing your sensibility to detect, but not the sensitivity to discriminate things. So in a sense, it's since I'm more prone to see things because I'm more excited, it's just a matter of bias and not really sensitivity. And people were discussing that. So since we had this design, we jumped and tried to see whether this anticipatory states, whether this pre-alpha oscillations, pre-stimulus alpha oscillations was modulating the stimulus representation in a second time after it was presented. So what was the task? The task was a really detection task, and there were two streams, one at the right hemifield and the other one in the left hemifield. It was interleaved. So people had to just attend to one hemifield, and it was dependent on the block, could be the left or the right hemifield. And they had to detect only when this a Gabor patch with a higher frequency was shown. And so when there was more lines in the Gabor patch, they had to just press the button. And actually, we're not really interested in the targets. We were more interested in the stimuli that were ignored by the participants, so they standards. And another thing is that all the stimuli, including the standards, they could have two orientations. So either vertical or horizontal orientation. And that's what we were focused on the decoding, and you are going to see right after. But first, we look at that alpha oscillations, and 0 is when one standard was presented, and here we see the frequency map, the difference in frequency map between attended and ignored stimuli, and you can see that for attended stimuli, you have less alpha. And well, here is the topography of this effect. So you see that is really, yeah, you have this contralateral effect. Well, another thing from this map is that here you use like a sliding window. So when I see that there is an effect before 0, it could be true. It could be because there is something before 0, but it could be just this is mirroring effect because I'm doing the sliding window. So in the next result, I'm just going to focus on a time window pre-stimulus, basically. So from 300 milliseconds until 0, and I'm going to estimate the power of those oscillations. And actually, I focus on more than my alpha band just to make the point that we had the effect only in the alpha band. So that's what we see here. So these are the frequencies that I estimated in that short time window before my standard stimulus. So you can see that it goes from 6 until 20 hertz, more or less. And the only frequency that is significant here is this one, almost 10 hertz. The other ones, so if you compare the frequency between attended and ignored stimuli, they are not significant in the other frequencies if you do like a correction, because I'm comparing a lot of times. So here the only that remains significant after the p value correction is the 10 hertz. And we also tried to see if we had this difference in how, if alpha oscillations were triggering this higher well, response time. So here what we see is we focus more on the alpha band. So for 10 hertz and 14 hertz, 13 hertz ish. So this, the first one is the probability of response since it was a target. So you can see that there is no much linear relationship between a low and high alpha power, neither for this frequency nor for a higher well, or beta frequency. And also there is, well, this is almost significant actually in 10 hertz, but it's not significant. And same thing for the response time. There is no relationship between how much alpha you have with how fast you are. So there is no relationship between those two variables. So far, so good. So we wanted now to see how my stimuli were actually represented in a signal. Can I decode the orientation that was presented, even though all those stimuli actually were irrelevant to the task, because they only needed to respond to those higher frequency Gabor patches. So here we see the decoding of the, well, orientation of the Gabor patch only for the standard stimuli. And those standard stimuli could happen in an attended M field or in an ignored M field. And you see that we can only decode the orientation when those stimuli were presented in an attended M field. So those bars here, blue bars, are the difference between 0.5, which is the guessing, because I had only 2 orientations. And you can see that there is also a difference between attended and ignored scores which are the bars, black bars over here. And this is also related to the behavior. So if we just gather how much, my decoding score for attended stimuli minus the ignored stimuli, and do a cross participant correlation, you see that so the participants that I think did better the task were also those with the G prime, were also those which we see a clearly difference in the decoding, in the feature decoding here. So actually, this is just a matter of proving a point that, the decoding here is capturing how well people are seeing, or how well the sensory, how well was the sensory processing of that stimulus. And the second question was, okay, so we see that we can decode in the attended, for the attended stimuli, the orientation of those Gabor patches. So let's focus on just attended stimuli and see whether in those trials, if it depends on the price stimulus alpha that you have in that trial. And these are the results for 10 Hertz and 13 Hertz. So you can see, here we divided the trials in two. We did a median split with trials with a high power and trials with low price stimulus alpha power. And we did the decoding at the same way. And we see that for low price stimulus alpha power, we see a difference actually not at the same time that we see for attended and ignored. Actually, it's much further away in time, so it's much later, for more or less from 300 milliseconds into 400 milliseconds. And what does it mean in the end? So we don't see the same thing for 13 Hertz. So we see that our price stimulus alpha power is changing how well is changing the sensory processing, and is changing how well the stimulus is represented in the signal. But it's not in a time window that we expected. It's much later. So in this sense, we kind of speculated that since it's more related to a later processing stage, could be related to how well this will, how well people will remember it, how well this could be, participants will be aware of that particular stimulus. But in a sense, the price stimulus alpha power is changing how well the sensory processing is doing, but not in an early time window. So you can see that it's not the same as we see for attended and ignored stimuli. And so for conclusions, we see that it's not only a bias, we see that price stimulus alpha power is changing the sensitivity, how well those stimuli are processed, but in a sense that how well is endured in the signal. So it stays there more time than when you have a higher alpha power. And I don't know if you noticed, but it's a kind of extreme way. So we were also interested in temporal processing. So how temporal attention was related in this task, this we couldn't see, because those streams are they are continuous, and at the time people were kind of criticizing this type of task, because you couldn't really measure what is entrainment from what is evoked responses. So we tried to go a bit further on this question. So since you know the timing and you kind of engage an entrainment with this type of task, but you cannot really dissociate what is driven by entrainment and what is just a matter of evoked responses. We came up with a different design where we do the entrainment, but then we put a silent period and see how the activity goes during the silent period, instead of just analyzing when you are presenting the entrainment stimuli. And we had a hypothesis that if you present the same thing a lot of times, maybe in this silent period, you would also continue to have this different representation. So you would create better representations when you are expecting the most the stimuli, compared to phases of the entrainment when you don't expect at all any stimulus happening, and so on for some cycles. And this is our hypothesis for temporal attention, basically related to strings or rhythm temporal attention, compared to just well, when you don't have at all, you just wait for the stimulus to appear 3 seconds after. So you just see the stimulus there, and then 3 seconds you have this bump in activity, and you see. So we wanted to test it because we didn't know whether this wasn't a hypothesis. We didn't know whether this is how temporal expectations or rhythm temporal expectations would work. And that's what we did in our next paper. So we had, in the next paper, we had 2 conditions. One, what was non-rhythm. So we presented a target, and after 3 seconds, we presented a probe, and it was a discrimination task. So the participants needed to say whether the probe, if it was in an auditory trial, whether the probe had a higher or lower frequency, and in the visual condition, whether the orientation was tilted clockwise or counterclockwise to the target. And in the rhythm trials, we presented the target actually a lot of times and in a rhythm fashion. So in the auditory trial, actually we had the 2 streams, the auditory and the visual stream, but we said, well, in the beginning of the block, we told participants to just pay attention to the auditory stream. So how was it presented? We presented a tone, and after 500 milliseconds, it was presented a Gabor patch with a certain orientation, and then after 500 milliseconds, again the same tone. So we presented the same tone a lot of times, could be from 3 to 6 times. And the Gabor patches, the orientations were the same as well. And across in the full experiment, we had 6 different orientations and 6 different tones. And in the visual trial was kind of the same, but people had to pay attention to the orientation, and the task was pretty the same as the non-rhythm. So after a while, we presented the target as and we marked the last one, saying, well, this is the target, so people would know that the following up would be the probe, the different one. And this marker was auditory trial, a higher volume. In the visual trial was a different color of the annulus. And then people would have to wait for the probe to answer whether it was a higher or lower pitch, and whether it was clockwise, counterclockwise Gabor patch. But we also have another thing here. So the probe could happen in different SOAs, and these SOAs were related to the stream that was presented. So the probe could be presented in 1, 1.5, 2, 2.5, and 3 seconds after the target. And so 1 here, 2, and 3 were in phase with the previous, with the previous attended stream. And 1.5 and 2.5 were out of phase with the attended stream. So in the sense, we had both. We had like this entrainment phase, and we have this silent period to understand whether we had these differences in pre-activation or expecting the feature, the expectation of this feature during the silent period. And this is again what I explained it. So we have the target. In the non-rhythm trials, the probe was 3 seconds after, and the analysis window will be just for the longer rhythm trials to be compared with the non-rhythm trials. So here we are going to select only the 2.5 and 3 seconds trials to analyze. So we would have a longer time window to understand whether we saw this fluctuation in those templates, basically. And for the behavior, actually, we didn't see any effect of this rhythm expectation, actually. We only see the result rate effect. So for trials, for longer SOAs, people were faster to respond compared to shorter SOAs. This was pity because we were expecting to have different accuracy and reaction times related to in phase, out of phase time spots, but SOAs, but not really. And so here is the expectation. So since we had 6 different orientations and 6 different tones, we could try to classify them in this silent period. And here 0 is when the target was presented, and here the probe could happen 2.5 seconds after. But we see that when we try to classify this very specific, this stimulus-specific information, we don't see anything occurring during the silent period. This is not what we expected from the hypothesis. So actually, it's not even a flat but significant classification. It's just flat, and well, we don't see any information during this silent period at all, neither for the tone decoding nor for grating decoding, neither for attended condition or for unattended condition. So this was a bit frustrating, but well, maybe that's not how temporal expectations work. So this is really stimulus-specific information that we are trying to decode during this silent period. And one thing that we thought afterwards, after doing the analysis, we thought maybe it's temporal expectations are not focusing on specific populations related to the orientation or tone coding, but it's more a broad generalized activity in the auditory cortex and the visual cortex. So then we focus on, well, in the silent period, can we decode the sensory modality that participants are expecting or attending to, expecting to respond? And that's what we did next. So here we have the non-rhythm trials, and in 0 we have the target presentation. So you can see that we can decode the sensory modality pretty well. And during this time window when there is nothing going on the screen, from 1 more or less to 2.5, you see that there is a not really significant, but in the end becomes significant again, the sensory modality that they are expecting to respond, or they are expecting at least the probe to appear. So it will be a visual or auditory probe. But what happens in the rhythm condition? In the rhythm, so here is the analysis window again. But what happens in the rhythm condition? So here in 0, we have the target. In 0.5, we have the other stimulus, because remember that if you are paying attention to the visual modality, it's a visual target, then you have an attended auditory stimulus. So here it is. But then you have this silent period with nothing going on. And you can see that we can clearly decode the sensory modality during this period. And this sensory representation is oscillating with the expectation that participants have in time. So you can see that there is a bump more or less in 1. Then 1.5, it goes again to the chance, basically, and in 2 it raises again. So you have this bias towards the sensory modality that you participants are expecting to see a target, so a probe, basically. And to see whether this is a real oscillation or not, we model this activity with a linear model, and here in red, and a cosine model with, well, actually 1 Hertz sine cosine model, plus, so it was nested, the linear model. So we could have this sine wave, but it could be shifted. So if we have had a difference as well in the y-axis. And what we see is that for rhythm trials, the sine model was way better than just the linear model, and but not for the non-rhythm trials. So this is a comparison in F values, and when we just compare the the amplitude of this sine part, the sine parameter. And one thing that we also can extract is, okay, so we see that there is a better fit for the sinusoidal model, but are those phases reliable, or they are the same phase for all the participants? Because we don't expect the phases to be random because of how we designed the task. And yeah, if you see the estimated phases for this model, well, the phases are distributed randomly in the non-rhythmic trial, but you see that there is a clearly preferentiation phase for the rhythm. So you can see that during 1 and 2 you have this hills, and in 1.5 basically you have more the valley. Yeah. And this is described here. So what are the conclusions? The conclusions is that this anticipatory states enhanced neural processing of the stimuli, and these anticipatory states are really, well, they depend on the attention mechanism that you are going to recruit, and to what dimension you are going to attend. And yeah, and attention to different dimensions seem to evoke different processing effects and mechanisms. So you could see that while in the spatial attention, you had this long-lasting representation because of the pre-stimulus alpha power, but in temporal attention, you don't see this anticipation in specific populations. So it was more a general sensory activation in a sense. And this brought us more questions actually, and that's what I'm working now. So one thing that, well, I presented here how people attend to space and how people attend to time, but you can imagine that there are different types as well, different dimensions. So how people attend to semantic categories. So yeah, I'm going to present, if you are interested in this topic, I'm going to present the results that I'm working on in my postdoc in the CNS Congress and ICON Congress. So if you are there, just pass through the poster and we can discuss more. One thing that we are doing at ONEHA and INT as well is to understand a little bit more the sense of agency and how sense of agency impacts the perception and impacts the sensory processing, whether you perceive it better, you have a better sensitivity, or if it's just bias your perception towards what you towards your prior action prior. And we also are working with eye movements, because I'm in a group that work with eye movements, and have a work, if you are interested in, related to decoding transsaccadic prediction error. And that's what I've been doing now. And yeah, I think I'm over in time, but I would just want to thank you, my previous labs and my funding resource, and well, all the institutions that were related to the projects that I worked on. And yeah, I think you have questions, right? So yeah, I'm open for it. Thank you very much.